last week, yeah, about a week ago, Wallace asked me to do the honor of doing a tutorial in 15 minutes on developmental biology. I was like, no way, I can't do that in 15 minutes. But I said, maybe I can do a quick one on morphogenesis. So he's like, okay, that's probably good enough. So I will try my best to introduce morphogenesis to you in 15 minutes. So I know a lot of you here actually are experts in morphogenesis, so bear with me for oversimplifying. But I guess the aim is to really introduce the field to people who've never really thought about morphogenesis before yesterday, hopefully. And really the idea is, well, what is morphogenesis? Is really the control of organism size and shape, right? And of course, we all know size is really important, and just look at the variations around us. But also, in my opinion, 3D shape or tissue architecture, tissue geometry, that's just as important, because of course we all know, the reason why our organs function optimally is really tied to the form they have in 3D, right? And just look at even just within humans, every organ is really specialized for a function, and often is really linked to their morphology, right? And so also another thing is growth, size and shape are separate factors. So for example, look at these wings. You can clearly see that there are different sizes, but I would say the proportions are the same in this kind of group. Whereas here you have different shapes, but you could argue maybe the sizes are more similar. And so in my opinion, morphogenesis is really about coordinating growth within the organism, but also across different species, I suppose. And how do you control size and shape of your organs in the same organism so that everything works in proportion and therefore optimally? And again, we've observed these proportionalities for really a long time now, and of course then we would like to understand really how is this all controlled. And for me, that's really one of the most fascinating questions in biology, the control of organ size and shape. And as a result, of course, of this, we see beautiful patterns emerging in biology, right? Often, lots of these, really beautiful patterns and functions and structures and linking, where I think biologists and mathematicians have really tried to coordinate that and link these fields already for hundreds of years. And so I guess with this workshop, we'd like to understand or try to inspire mathematicians to think about morphogenesis maybe in new ways, using methods they're familiar with, and hopefully inspire us, biologists to think about new ways to analyze the data we have and the cell dynamics and so on. So I want to focus today really, again, just reiterating the concept that development or morphogenesis really, everything starts from a single cell, right? How do you grow from a single cell to millions of cells? So even just taking the Drosophila, which is a commonly used model organism to look at morphogenesis. So for example, again, just looking at the wing, which is obviously critical for the flight and function of the fly. The wing is always of a certain stereotype size and shape in wild-type systems, and therefore the growth and the morphogenesis is a very tightly controlled process that many labs across the world look at. But hopefully you appreciate, there's huge amounts of growth in terms of increase in cell number over just the course of a few days, but also changes in 3D morphology to, in the end, get to this, tightly controlled structure. So there's huge amounts of growth and increase in cell number needed. That's a very, basic requirement. But another feature is, of course, this is not static, right? It's very dynamic processes. So even in just this movie, hopefully you appreciate there are cells dividing into two. That proliferation, that increase in cell number is critical for growth of an organism, right? But also, hopefully you can see cells are always moving, changing neighbors dynamically. So everything is a very dynamic process. So again, we want to understand this dynamics. How can we model this dynamics? How can we study these, this dynamical data using different mathematical ways? So I would like to conceptually break this down a little bit, therefore. So yes, how do we go from a single cell to a beautiful structure in 3D, right? And sorry if I can actually progress the slides. Yeah. So I wanted to break it down as sort of simply as possible into these, in my opinion, 5 basic steps. It's not exhaustive. You can think of other ones, but in my opinion, these are the 5 basic steps that are necessary to grow a structure. And they can happen, of course, in parallel, but again, for the sake of this tutorial, I will talk about them linearly. And so in my opinion, what you need to do is you have to grow your material, right? How do you grow material to the right amount of material and then stop? And so most of the time, it's really to do with increasing cell number, because there's a limit to how big a single cell can get to. So if you want a big organism, it's really through increasing cell number through the control of growth of cells and the division rates of cells, and equally death rates of cells. Another way is to actually control division orientations. So whenever a cell divides into two, it can divide that in different orientations, right? So we'll go into these details later. So that's already another way to control the direction of growth, right? Just by controlling division orientations. A third way is, imagine nothing changes in terms of cell number or cell division rates, but just simply the cells change their shape. They can become sausage shapes. Already that is sufficient to elongate in one axis, right? Or, for example, we're all cells here. We start to move, intercalate, we change our neighborhoods. We start to flow relative to each other. That is another way to change the shape of this collective massive, population of cells, right? And then finally, these are all, I would say, so 2D systems. You can think about this in 2 dimensions, but of course we are 3D systems and structures. And so how do we get beautiful 3D architecture? For example, outplane folding, which is the first basic symmetry breaking event you need to have. And so again, all of these processes, in my opinion, I've sort of tried to break them up into simple components, but that is really the take-home kind of message here. So if you leave this tutorial with nothing, but just try to remember this slide, and that's probably all you need to think about for today. All right, but I'll go through each of these a little bit more in detail just to give you some ideas of how these are controlled, what we know, and how can we use new tools now to understand these processes. All right, so growth rates. So to make it as simple as possible is really about controlling the cell cycle, right? So in the cell cycle, there are phases where cells grow their material, double their mass, DNA content, and then when it's right timing, they divide into two through process of mitosis. And so this has fascinated biologists, I think, for really decades. I mean, this is fundamental to so many things. For example, when the cell cycle is misregulated, you have disease such as cancer in the simplest, but of course, many other problems. And so there's really hundreds, I would say hundreds and thousands of genes and biochemical signaling pathways that are now understood to feed into different phases of the cell cycle to either speed up the phases or to shorten them, and also to regulate the proportions of time that each cell spends in each of these phases, which in the end will really change the dynamics of the whole system. So I won't dwell into that because again, that could take me years to go through, but just to remember, there are lots of biochemical signaling pathways, GRNs, people are modeling, gene networks, all of these will feed into cell cycle regulation and in a way will control growth and proliferation rates. So that's one way to control how fast and how big you grow to, okay? And again, I'm sorry if I'm oversimplifying this, is really to just give you a quick snapshot overview of what we know. But apart from these genes and signaling pathways, there are also other ways to control cell proliferation and growth, which actually is through mechanical forces. So it's really amazing, like about 20 years ago now almost, people realized that actually if you grow cells on different stiffness environments, for example, either on glass or very soft gels, these cells actually will somehow react to this physical environment. They can sense the stiffness of the environment. As a result, they will change their cell shape. And as a result, they will actually cause different signaling molecules that I talked about earlier to shuffle in between the nucleus or out. And as a result, this can cause the cells to divide faster or slower. So to give you an analogy, so imagine if I'm a single cell, I'm trying to represent this one here. I'm grown on a very stiff glass substrate. So I end up being stretched and pulled and really extended in my shape. And as a result, I actually start dividing more because intuitively, I don't want to be stretched and stressed. So I want to divide more to kind of relieve that stress. But the opposite is also true. If I'm growing on a very soft kind of gel, like, custody gel kind of environment, then I start to kind of contract up and into this kind of right side phenotype. Lots of signaling pathways are changed. And as a result, I stop dividing. I go into some kind of, pause in growth and division. So the simple kind of take-home message here is that actually, even if all the signaling pathways and growth factors I get are the same, but just by being in a different mechanical environment, cells can change their growth rates, which again is pretty cool if you want to factor those into your models and so on, right? But of course, the total net increase in cell number is not just how fast cells divide, but also how fast they die, right? It's that balance that's important. So cell death is also actually a very tightly regulated process. So for example, why I have 5 digits is because cell death is controlled in certain locations. Otherwise, I'll have more or less digits, which of course is a problem. So there are many parts of our bodies where cell death is a tightly controlled process. So for example, in the gut, the cells are mechanically almost squeezed out. So just by feeling this environment and almost the physical kind of factors, cells can be squeezed out to regulate total cell number and the shape of the organ, and so on and so forth. So yes, again, there are signaling pathways that will actively decide which cells should die, but also sensing the whole physical kind of compactness of the environment can be another way to induce cell death in a very controlled way, so that cell number is tightly tied to morphology and the physical state of the organ, right? So another thing is cell division orientation. So again, for now, I've kind of assumed cells are dividing randomly, but of course, if you divide randomly, probability will ensure that you'll turn into a sphere, but most of us are not spherical. So how do we change that? And in fact, there's been observations now for almost, 150 years, people have known that it's really the geometry of the mother cell that pretty much dictates how the cell will divide. I.e., if you have a long cell like this, it will almost always divide to cut the cell into 2 shorter cells. Otherwise, you will have, weird cell shapes accumulating. So this geometry really dictates division orientation, and this is observed in almost all organisms across. And so already that tells you we can tie in geometry to predicting divisions. And again, that should be part of the models that we build, right? Another thing, as I said, is, well, just simply changing shape, right? Clearly, again, if I just stretch myself or squeeze myself, I change how I look, right? But that's easy in a way to think about for a single cell in isolation. So for example, if you focus on the single cell in the middle, this orange cell, it can adhere to neighbors to stretch, or it can also contract in a polarized way, for example, through polarized ectomyosin structures. This will make a spherical cell into anisotropic shapes, right? But in multicellular systems, this middle cell, apart from its own cell shape control, will also be influenced by neighbors. It can be squashed by neighbors growing into it. It can be stretched by neighbors pulling on it. So then actually control of a single cell shape in a multicellular context is a very complicated collective dynamic behavior, which again, I think is very hard intuitively for biologists to think about without kind of trying to create models and predicting these behaviors, right? And so how are these global forces generated? So one simple way could actually be through differential growth. So this is a simulation of a vertex model that I made when I was actually still a postdoc. And the aim of this, hopefully you can appreciate, is that you can see these cells in the middle growing and dividing. So every time you see a kind of a balloon, blub, blub, blub into 2, this is cell division on the screen. And as a result, I just want to emphasize nothing at the cellular level is different, right? These are all the same, but just by these cells growing and dividing faster, hopefully you can appreciate these surrounding cells are changing shape, right? They are being stretched along the periphery because there's increased pressure here, pushing on them, compressing them, and these are passively responding and stretching. So what I'm saying is here, there's no single cell level anisotropy or heterogeneity, yet this emergent behavior arises just simply because of one rule, that these middle cells are growing faster and dividing faster than the other cells. So then if you think about this in sort of, if you have this heterogeneity distributed across, you can really sculpt whatever shape you want just by controlling growth rates. And this passive secondary effect of cells acting on the neighbors will really reinforce the growth dynamics and so on and so forth. So there's actually complex feedback in the system. So we can talk about that more if we want to. But again, so this changes shape. If we then think about 3D, the concepts are exactly the same, right? So instead of just all of these forces I talked about acting on the apical surface of a cell, it can also act laterally, it can also act basally. And if they are different, apical, basal, you will get indentations or invaginations, out of place symmetry breaking events. So again, that concept can be extended to different dimensions, and it's the same principles. And so then one last few things is, what about cell rearrangements, right? So again, before all of the above three processes assume that cells stick together, but in fact they don't. So again, just look at this difference. You can stretch a patch of cells by just stretching the cells, but the red are touching the red here, right? But also they can move and change neighbors. So in this case, now the blue cells are no longer touching each other because there's been a dynamic rearrangement, what I call actually cell fluidity, tissue fluidity. Cells are flowing past each other. And this is another way to stretch this patch of tissue. So you get the same outline, but the cell topology is now actually different. The connectivity is different, right? So that's another way animals and plants, but mostly animals, organize their control their shapes. And so, for example, we actually found a few years ago that upon wounding of an epithelium, so here we kill these cells with a laser, and we use this to analyze how does the epithelium repair itself. And we found an amazing new mechanism where the cells actually undergo a phase transition. They go from a solid state, i.e. they're normally stuck together quite happily, homeostasis, you want to stick together. But then when I punch a hole, the cells around the wound start to flow relative to each other, like molecules of water. And hopefully you appreciate, as the wound closes, you see some of the cells actually have left the wound edge, right? They all started touching the wound edge. That's why they're blue. But in the end, by definition, the wound edge is a dot, and there are only 3 cells there because everything has rearranged itself, moved itself. And so analogy we say is there is basically a solid to liquid transition. And what is the easiest way? For example, if you poke an ice cube, how do you fill the hole in ice cube? You melt the ice cube. The water will flow into the hole. That's the most intuitive, simplest way, in fact. And that's kind of actually amazing. We found this out only a few years ago. But I think this analogy really, again, is to do with cell topological changes and so on. Okay. And then lastly, 3D sculpting. So of course, we need to bend our flat sheets into different structures. And in the real organisms, there are lots of folded structures. But in fact, every branch starts from the out of plane fold, right? And again, many labs have tried to study this. I won't go into this in too much detail, many mechanical models, but also there are different ways to generate folds. So for example, you can have just differential growth rates. Again, in this case, you can have cells being stretched in 2D, but also the cells can cope with the folds by bending in 3D. And so again, by just controlling growth rates, you can control different stresses building up. And in 3D, this will generate folds in certain positions. So again, we showed this with the finite element modeling approach that we did before. So I guess what I would like to really, sort of the aim of this tutorial is to hopefully find new ways to model these behaviors. There's lots of mechanical models out there, but are there other ways to study this beyond the GRNs and the mechanical models? And can we try to come up with some new ideas in this field? All right, thanks very much.
Thank you. We have time for 1 or 2 quick questions, if there are any quick questions. Yes.
I was wondering here, actually, I'm glad we're on this slide. So you're showing a lot of rearrangements and stuff happening, through these fluid interactions. But do we typically form our 3-dimensional structures from layers of planes?
We also do. Yes, we do. Okay. We do also. So obviously, I was still trying to simplify, for example, skin is multi-layered, the brain actually is multi-layers, and in fact, a lot of the folding structures, I think there was this, this here, one classical mechanism for how to fold is if your different layers actually grow at different rates, then the only way to keep the layers still intact, you have to fold, right? So yeah, so absolutely, many things are different layers, but I try to simplify it, but absolutely, yes.
Thank you.
Good. We had a second quick question at the back, yes.
When it comes to growth rates differing in space, you mentioned the case when the cells grow quicker on the inside, but a lot of the time I think it's the case that they actually are driven by the interface, the outer interface. And so I was wondering if there are any specific morphological changes induced by that.
Yeah, so that differential, that was a simple analogy. If you have more in the middle, and it was the computer simulation that, and although in fact lots of things grow like that, plants, meristems, the wing disc, for example. But for example, this is us trying to simulate wing disc. it's definitely not always, in this case, the middle is growing fast here, but the outer edges also all have differential rates. So I think, I guess what you're asking is the short answer is there are many different ways of regulating growth differentials, sometimes outside, sometimes, and they all occur. And as a result, that will really drive the dynamics of the system, but also the viscoelastic properties of the cells, how they cope with that differential, that will again determine how they fold, how they buckle. So I can't give you a concrete example because there are so many, but even here we've measured and the patterns are very complicated. There's hardly ever a sphere with the middle growing faster.
Thank you. Let's thank
All right,
one
okay.
more time.